Have you ever pictured your body as a massive, bustling factory? Because that's pretty much what it is. Inside every single one of your cells, there's an assembly line running 24/7. And it's one mission: to take the genetic blueprints, our DNA, and turn them into the tiny machines and structures that make life possible: proteins. So let's pull back the curtain and take a deep dive into this incredible molecular assembly line, a process we call translation, and see how it builds life, one protein at a time.
All right, so here's our game plan for this journey. First up, we're going to see how the factory itself actually gets built. Then, we'll figure out how to read the instruction manual. After that, we'll meet the specialized workers who bring in all the raw materials, and then we'll see the assembly line going at full tilt. And finally, we'll look at how the factory handles mistakes and even deliberate sabotage.
So let's start where it all begins. You can't build anything until you have the machinery to do it, right? And this brings up a really fascinating, almost chicken and egg kind of question. How does a cell build the machine that's responsible for building pretty much every other machine it has? The answer lies in constructing the heart of our factory, the ribosome. This whole process is just a beautiful example of cellular logistics. If you follow the diagram, you'll see that the main parts for the ribosome, this stuff called ribosomal RNA or rRNA, are made right here in a special part of the nucleus called the nucleolus. But the protein components are actually built way out in the cytoplasm. These proteins then get shipped back into the nucleolus, where they meet up with the RNA and get assembled into the two big chunks of our factory. Let's quickly break down who the key players are here. The RNA is like the steel frame of the ribosome. It provides the structure. The ribosomal proteins are like the nuts and bolts that give it stability and help it function. And then you have these things called SNO RNAs, that's small nucleolar RNAs, which act like tiny precision laser cutters. They take this big initial sheet of material, the 45S precursor, and snip it into the final, perfectly sized RNA pieces. It's incredibly efficient.
Now, you're going to hear scientists talk about these 40S and 60S subunits, and that S stands for Svedberg unit. And it's a little weird. It's a measure of how fast something sinks in a centrifuge, which has to do with both mass and shape. The crucial thing to remember is it's not simple math. That's why when you put a 40S and a 60S piece together, you get an 80S ribosome, not an 100S one. Think of it like two Lego pieces with complex shapes. When they snap together, the final object is more compact than you'd expect. Okay, so our factory is built, the machinery is humming, now we need the instruction manual. And that comes in the form of something called messenger RNA, or mRNA. Let's see how the factory actually reads this genetic blueprint. This table you're looking at is basically the Rosetta Stone of life. It's the dictionary that translates from the language of genes to the language of proteins. The mRNA is read in these three-letter words called codons. Each codon, like GGU or AUG, tells the ribosome which one of the 20 amino acids to grab next. The assembly line always knows exactly where to start thanks to the start codon, AUG, and it knows exactly when to stop when it hits one of the three stop codons. And what's just amazing about this genetic dictionary are two key properties. First, it's universal. The codon ULU means phenylalanine in you, in a bacterium, in a fly, in a banana tree. It is the shared language of all life on Earth. Second, it's degenerate, which is a fancy way of saying it's redundant. As you can see, multiple codons can code for the same amino acid. This is actually a brilliant design feature. It acts as a buffer against mutations. A small typo in the DNA might not even change the protein in the end. But here's the catch. How the ribosome reads these three-letter words is absolutely critical. This is what we call the reading frame. Just look at the example here. If you start reading from the first letter, you get one specific protein. But if you shift over and start reading from the second letter, the groups of three completely change and you get a totally different, garbled protein. That's why the start codon AUG is so important. It sets the starting point and makes sure the message is read correctly from the very beginning. So we've got the factory and we've got the blueprint, but how do we get the actual building blocks, the amino acids, to the assembly line at just the right moment? Well, for that, we need our molecular matchmakers, the transfer RNA or tRNA. In textbooks, you'll almost always see tRNA drawn in this flat cloverleaf shape on the left. It's easy to look at, but in the real three-dimensional world of the cell, it folds up into this compact L shape that you see on the right. This molecule is a perfect adapter. Down at one end, it has an anticodon that's designed to read a specific codon on the mRNA. And up at the other end, it carries the one amino acid that corresponds to that codon. It's the ultimate delivery molecule. Now, before a tRNA can do its job, it has to be charged with the right amino acid. It's a quick two-step process. First, the amino acid gets a little energy boost from ATP. Then, a super-specific enzyme, get ready for this name, an amino-acyl tRNA synthetase, acts like a master craftsman. It recognizes one specific amino acid and its one matching tRNA and links them together. The accuracy here is just phenomenal.
And this is a little look under the hood at the chemistry. The main takeaway here is that the cell spends a bit of energy in the form of ATP to create a high-energy bond that hooks the amino acid onto the tRNA. That energy doesn't just disappear, it's stored in that bond. And that stored energy is going to be the power source that drives the whole protein assembly line forward.
All right, this is it, the moment we've been building up to. The factory is online, the blueprints are being fed in, and the delivery trucks are all loaded up. It's time to fire up the assembly line and watch how a protein actually gets built, step by step.
So, the ribosome has three main workstations, or sites. Think of them as docking bays. There's the A site, the P site, and the E site. The A site is for acceptor, or arrival. That's where the new tRNA docks. The P site is for processing. That's where the growing protein chain is held. And the E site is for exit, where the empty tRNA gets ejected. The easiest way to remember it is APE, arrival, processing, exit. Let's watch the cycle in motion. Okay, step 1. A new tRNA carrying the next amino acid zips into the open A site. It has to be a perfect match for the mRNA codon waiting there. Then, step 2, and this is the magic moment. The ribosome itself causes a peptide bond to form, and the entire growing protein chain gets transferred from the tRNA in the P site over to the new amino acid in the A site, just like that. What happens next? Well, in step 3, the whole ribosome moves. It just chunks slides forward, exactly one codon down the mRNA track. This moves the tRNA that was in the A site over to the P site, and the now empty tRNA from the P site shuffles into the E site. And just like that, the A site is wide open again, ready for the next delivery. Finally, step 4. The used empty tRNA in the E site gets kicked out, ready to be recharged and do it all over again. Now, this is all happening incredibly fast, so you might be wondering, how does it stay so accurate? Well, it has help from a couple of molecular supervisors. First, you have a factor called EF2. Think of it like a bouncer at the A site. It double-checks the match between the new tRNA and the mRNA. If it's not a perfect fit, it gets rejected. Then, after the chain transfer, another factor, EFG, helps the ribosome slide forward smoothly and make sure the whole line doesn't get jammed up. So how does the assembly line know when the protein is done? The mRNA blueprint has one of 3 special stop codons written into it. And here's the thing, there are no tRNAs that match these codons. Instead, when the ribosome hits a stop codon, a totally different protein called a release factor binds in the A site. It's like the foreman hitting the big red emergency stop button. This triggers a reaction that cuts the brand new protein chain free, and it floats away to do its job. The whole factory then falls apart, ready to start a new project. Okay, so the factory has built its product. Amazing. But our story isn't quite over. We need to zoom out and look at the bigger picture of quality control and what happens when this elegant process gets sabotaged from the outside. At first glance, these two ribosomes look pretty similar, don't they? On the left, you have a bacterial ribosome, and on the right, one of ours, a eukaryotic one. But there are tiny, subtle differences. Notice the bacterial one is a bit smaller, a 70S unit, while ours is 80S. It's also built from slightly different parts. This small difference might not seem like a big deal, but it's a crucial vulnerability, and it's one we can exploit. This is exactly how many of our best antibiotics work. They are precision weapons designed to target those small differences. Let's take tetracycline. It's shaped perfectly to wedge itself into the A site of a bacterial ribosome. It's like jamming a block of wood in the docking bay. No new tRNAs can get in, protein production grinds to a halt, and the bacteria die. But here's the brilliant part. It doesn't fit into our A site. So the drug can kill the bacteria while leaving our own cellular factories completely unharmed. But what about mistakes that happen on the inside? Sometimes a cell makes a faulty mRNA blueprint, one with a premature stop codon by mistake. If the factory made this, it would produce a broken, potentially toxic protein. So the cell has this amazing surveillance system called nonsense-mediated mRNA decay. Basically, the cell leaves little protein markers on the mRNA during processing. If a ribosome hits a stop signal before it has passed all the markers, alarm bells go off. The cell flags that blueprint as defective and calls in a cleanup crew to destroy the mRNA immediately. It's incredible.
When you step back and look at it all, from building the factory to reading the blueprints to proofreading the assembly line, translation is just a symphony of molecular precision. And this assembly line is running countless times every single second in our bodies. Which leaves us with one final big thought. With all this activity, what are the real consequences when one of those quality control systems fails and just a single faulty blueprint slips through the cracks? The answer to that question is at the heart of countless genetic diseases.